Next up, we have Dr. Michele Caggana. She's joining us remotely today. Dr. Caggana is the director of the New York State Newborn Screening Program and also the co-project director for NIMAC. Welcome, Dr. Caggana.
Thank you. I'm sorry that I'm not able to be with you in person. Had a couple things happen. My son is a senior and went to the state soccer championships, and last night was their big high school banquet. And tonight my husband's getting an award for work, so kind of two personal things interfered with work for once. I hope everyone has enjoyed the session. I've been listening in quietly, and the talks have all seemed were very good, and it's really nice to hear all that information. I'm going to go over today information on second-tier molecular testing for both Pompe and MPS, and we've heard snippets about molecular testing, and what I wanted to do in this talk was actually to go over what it takes to set up a test and some information on why we bother to do it in the first place. So thanks, Beth, and hopefully the slide switched. I've asked Scott to throw things at people that fall asleep since I can't see you. So here we go. What types of molecular? There, now I know you're all awake. What types of molecular testing are actually available? So one of the things that we look at is whether or not we just want to perhaps look at these pseudodeficiency alleles that we talked about and you've already heard about. So you might be able to set up a molecular test just to look at those and rule those kids out. Another thing that you might think about doing for these conditions, both Pompe and MPS, we've heard that there are some common mutations that occur in both of these conditions, and maybe we could set up a targeted molecular panel and we could take a look at just those mutations and see whether or not, and it would be similar to what we do for cystic fibrosis, where we might look at a panel of just a few targeted mutations. We'd have to probably set that up on our own because there's not a big vendor push to create a panel like that. The next thing that you could think about is actually sequencing the gene for Pompe, the GAA gene, or the IDUA gene for MPS 1. And that's what we've chosen to do, mostly from precedence with what we've done with Krabbe disease. The next thing you might think about as you move along the way here is you can set up a next-gen type test. That's on a fancier instrument. I have a picture of one of them for you later. Or you can also set up the whole panel of LSDs. So we've heard from Illinois, Missouri, they do multiple genes. And maybe you want to set up a next-gen sequence for all of those genes, and then you only can take a look at the gene that you're interested in. So that's the direction I'm kind of headed as we move forward with adding these other types of conditions to the screening panel. But right now, for today, we're just doing the sort of old-school Sanger sequencing. So I like to think of this as sort of a triangular approach. So we started off in newborn screening at the very bottom, and most programs do some sort of genotyping of some panel of mutations. And then some of them do single gene sequencing. And then you can move to do Sanger sequencing of panels or next gen. Then you can even set up, some companies are even setting up sequencing of the entire spectrum of newborn screening genes. And then you can just do the whole darn exome and see whether or not what you can reveal about these babies. And people talk about this a lot, but it's sort of pie in the sky for a newborn screening program. So the future, I think, is moving in that direction. I don't know if I'll see it in my career, but perhaps I will. And of interest, when I Googled pictures of the future, there's actually a rapper who's called The Future, and he came up more often at this time. So it was kind of an interesting exercise for me. My son obviously knew of him and thought I was an idiot for not, but okay. So why do we even bother doing this? If you have the technology in-house and you have expertise in-house, people talk about how expensive it is. Our cost of newborn screening, we do everything on a shoestring budget, so to speak. So when you actually cost out, when you're only looking at a very small percentage of babies that go to DNA testing, Our costs are very different than those in a commercial lab that's paying a lot of infrastructure and marketing and billing staff, which we don't have any of those. And so our costs are actually quite lower than they are if you look up a list price from a commercial lab. As we already said, the pseudodeficiency alleles really help us to rule out. We've seen this a lot in Pompe. I think in MPS it's going to be very similar. The turnaround time. I'm going to, as I walk through how we do this testing in our laboratory, Aaron Hughes, who's done a great job with Pompe in our lab, has given me sort of how much time she spends hands-on and how much time things take hands-off. And we do some pre-prep work that helps us cut that time down, so we actually can get these results out in a little over a workday. Our doctors actually like the information, I think. I don't know if any of them are there. When we give them the genotype, if they know it's one that they need to be urgent about, they know they need to be urgent, and they take that and run with it rather than pondering whether it's a referral or not and what they should do. It also enhances how they interact with the family because they know how to deliver the message. If they're sure that the baby's condition, like we heard in the earlier talk, where we know these kids are sick even though they look well, they can relay that message a little bit better. The other thing that in public health we always are concerned about is healthcare disparity. So in families that are uninsured or have poor insurance, we give that information along with the newborn screen, and they don't have to worry about being able to pay for the sequencing test for later on after the fact. The other thing that Joe talked about, the cases of premature infants, we can release those babies if we have molecular results from any further workup. Or we could refocus the effort and say, maybe this child is sick and hypotonic and has a big tongue, and that's a problem, and be able to follow those kids a little bit more easily. So that's sort of why we actually embarked on this actually for Krabbe disease. And we found that by doing the screening test, we reduced referrals in Krabbe about 42%, and somewhere between 35 and 40%, I believe, for Pompe. Our numbers are still a little bit small. But between pseudodeficiencies and neutral variants, we've been able to reduce actually the number of kids that need to come in for any sort of follow-up. So I have here history in the molecular lab. On the top left side of the screen, that's how I learned how to do sequence back in the, I don't know what decade, probably the late 80s. We used radioactivity, very low throughput, and we had to manually read these gels. These were very long gels. And each of those 4 lanes that you see is one fragment from one person of DNA. You had to sit there and handwrite, have somebody read to you, and then sit there and handwrite or type to a computer what each of those bases were. Very slow. If we did the Human Genome Project this way, we would still be sequencing the human genome. That is called Sanger sequencing, and that was the topic Sanger sequencing, and that was described actually all the way back in 1977. As we move to the right on the screen, you will see a series of colored spikes. Those peaks are what we look at now for Sanger sequencing, and they're fluorescent labeled. So we get a whole piece of paper with anywhere from 600 to 800 bases on it, any individual person or fragment. And this is how laboratories that do Sanger sequencing now do that task. They don't use the radioactivity anymore. And then that nice jazzy box there on the bottom right, that is a whole exome type machine. And what this machine does is basically take the DNA from and you chop it up into a lot of pieces to make what we call a library. And it can sequence billions of pieces. And you could actually multiplex people on one instrument, and you get millions of reads. And so what it does is uses a bioinformatic approach. On the top left, very manual approach. The bottom right is very computer-driven approach to assemble sequences and get base reads. And there's a lot of laboratories in the diagnostic world and in the research world that are now using those types of instruments. Okay. So I put this up, Joe showed a a version that had all of his part of the assay up top as well. This is the DNA focused part of what we do for Pompe disease. And you can see what types of results we get in molecular end up resulting in referrals. And then you can see where we release those babies and they have no further workup. You've heard the term probably variant of unknown significance, or boost people call them, or bust people call them. That's when we get a base change or a DNA change and we really don't know what to do with it. So we nicely label it a variant of unknown significance. And these are the things that are problematic. In a clinical laboratory, you're getting a bias because you have a baby who's symptomatic or a child who's symptomatic, and the doctor thinks they have this, and they're actually looking for something to sort of confirm their clinical suspicion. And in newborn screening, we're coming from the other direction, where we have babies who look perfectly healthy, and we get these variants, and it's very, we do a lot of hand-wringing in trying to figure out what to say about these variants. And there's tools that we use, but it's difficult. When we talk about polys or polymorphisms, they're common variants that are not disease-causing. So those are the non-pathogenic variants that we are on this slide and we talked about previously. The pseudodeficiency alleles are shown at the bottom of the slide. It's a glycine to serine change and a glutamate to lysine change. These are the ones that cause the main pseudodeficiency that were reported out of the Taiwan study for Pompe disease, and we've found these in the New York population as well. So the overview of how we do a DNA assay, when we do a sequencing assay, we take 2 punches. We do our testing in duplicate. So we take 2 3mm punches and extract the DNA. And then for Pompe, we amplify 14 different gene fragments, so different parts that cover the entire gene, and we amplify them by PCR, the polymerase chain reaction. Those 14 fragments cover the whole coding region of the GAA gene and what's called the intron-exon boundaries. If we do linear sequence in the genome, there's pieces called introns that get spliced out or taken out when it's made into RNA before protein. And so it's very important to know what's at those boundaries, because mutations, even a little bit in the intron, could be problematic, because then the RNA is not made properly and the protein is not made properly. So anybody that does this type of sequence will look at the coding regions and then the exons, and then also a little bit in the introns on either side. We take those PCR products and then we use them for Sanger sequencing, which is the fluorescent type. And those sequences are then analyzed for mutations or variants. So step 1 is punch on the upper left side there, and then Erin is nicely holding for me yesterday a plate with specimens that she was extracting DNA from. And you can appreciate when we extract the DNA, we get basically trying to get rid of as much of the heme as possible in the blood spot. You can see one's a little pink yet, and the other ones look pretty white. So when you do this on the bottom, you can see that the hands-on time is actually only 5 to 10 minutes. There's some spinning and heating and shaking and other things that Erin can walk away from while they're happening. And so you can appreciate, while it might take an hour to extract DNA, it only takes about 10 minutes of hands-on time. This is just a layout of how we set these up. So the first thing we do is go to the databases, and then we get the gene sequence, and then we try and come up with how we're able to sequence this entire gene in one fell swoop. So we have these 20 exons, the PCR number and the number of fragments. So as I said, there were 14 of them. The GAA gene is on chromosome 17, and the protein has 952 amino acids. So the first step is there's programs you can that are free on the computer that you can help design primers for. So they sit on each side of the piece that you want to amplify, and they're sort of the clues for the whole assay to amplify that little piece of DNA. So you can see sometimes a PCR is only 1 exon covered, and then other times, like for PCR number 4 or 3, you got exons 4 to 5 covered, 6 to 8 covered. So they're different sizes. So we designed something that actually will enable us to do what we need to do in a way that we can do everything at one time. And it takes a little bit of practice and upfront work, and I have a little bit of a work in progress for MPS, which is what we're currently working on. This is the assay we use right now for Pompe. We design our primers, then we make sure that there's no base changes that sit underneath those primers. We try and use the same PCR program, and we can use different mixes to make all of these different cocktails to amplify the entire gene. So this is an example of what we call a plate map. I don't know if you can see my cursor. Can you see it? Okay. So the E is the exon, and 1A is punch number 1 and punch number 2. So we set these up, and this is a PCR plate. There's actually 96 wells on this plate. We also set up what's called a wild type control or an extraction control. And then this here is a no template control. So this is just water, and this helps us look at contamination. This helps us make sure everything's working properly. And you can read through here, and there's various, the way that they all have been set up for the assay. The way we do this is we set up the PCR plates. Those 96 well plates are set up in batches. So we use a system called Janus. It's these are pipette tips here, and they move liquids around. So each of these is one of those PCR plates I just showed you that map of, and we can prepare these in advance. So we do these because it saves us time. Instead of setting up everything individually every time we have a test to do, we just can pull a plate out of the freezer and use it. And then we can make these plates up in our spare time when we're not doing the sequencing assay. By batching it, it's better for quality improvement in QA and QC. Minimizes pipetting errors. I'm sure even if you're not in the lab, you can appreciate what it must be like to pipette multiple liquids into those little wells and prone for error. And it also helps minimize inconsistencies because we make up 10 at a time. So we have a 10 plate setup in each batch. We set this up for the PCR, and then we also set it up for the sequencing part of the assay. So when everything works well and we set up our reactions, we then run what's called an agarose gel, and this lets us check our products. So here we have exons 2, 3, so we got PCR 1, PCR 2, 3, 4 from the other slide. And you can see that everything looks pretty good. Everything amplified is pretty clean. And this lets us know, okay, we got the right piece amplified because we know what we're looking for, that everything worked, and that importantly there's no contamination. Each of these three fragments here, you'll see next to it, there's no band next to it here, and that means that we don't have any cross-contamination, which would be a problem certainly to do this, and we'd know we need to go back and start over again if we saw bands in those empty wells. Again, the time on and time off, hands on, hands off is shown at the bottom, and I have a summary at the end for you. So once we do the amplification, and when we actually do this, we use what's called a touchdown PCR, and that means the program actually decreases in temperature as it runs through the cycling, and that's what enables us to amplify everything on one plate at one time using a single set of conditions. The next thing we need to do is clean up the PCR products. So a couple hours have gone by. We take our PCR products, and now we set up what's called an exosap. Exosap is short for exonuclease and shrimp alkaline phosphatase. And what this does is exonuclease cuts away all the extra primers hanging around, and SAP takes off phosphates, so it gets rid of nucleotides that are hanging around from the PCR. And we just set this up on another plate. We put the PCR products in here, and we add the exosap to each well. And then from there, we put it into the machine again and let the enzyme do its work. At the end of this process, now it's cleaned up. Again, it's about 30 minutes in the thermocycler to do this, and a couple minutes to create that plate for you, to create that plate. The next step is actually the sequencing piece. So now we've amplified our gene, we've cleaned up the amplification products, and now we're actually going to do the sequencing. And we actually do this the same way we set up plates ahead of time on the robot. And so we're just going to go ahead now and add, there's new reagents in each of these plates, and we add the product that we just cleaned up into each of these wells. This is all done on the thermocycler, or sorry, the robot as well. Some of our products are quite big, so when you see something that says seq, that's just an internal sequence. So we do from one end in the other forward direction, and then you can see here these are R's, the reverse direction. So this is one case, all of this for just one case. We have the duplicates and all of the sequencing products. So we let the machine run. The sequencer takes a couple hours to run. That's a picture of the sequencer. It's kind of boring looking. It's just a box, and your plate goes in here, and it does the reactions here. And the computer looks pretty cool. It's got all the colors, so it's running through. This is the fluorescence actually running through the capillaries, so they can actually monitor as it runs to make sure everything's working okay. After we do the cycle sequencing, we clean it up again. We run it through a column, and then we actually load it up and let it go. So a lot of times they'll get all the way to here and then set this up to run overnight, so then the first thing in the morning they can take the sequence off and take a look at it. After the results come off, we analyze it using a computer program it helps us call mutations easily. Anything new, we have to figure out where it is, what the base change is, well, we know the base change from the sequencer, we're going to figure out what the amino acid change is. And then we can go ahead and search databases in the literature for mutations. There's also computer programs to run changes through to help us determine whether or not a variant that's new or never been reported is pathogenic. Again, that's information that the physician would find very helpful, and we have to do our best to make some sort of prediction about what we found. And then finally, we make the referral, and we hopefully have the best interpretation possible at that time, even though, as I said earlier, we really don't have any clinical information about this child at this point, because the whole point of why we do it is to have them healthy at the point we let them know that there's an issue. This can take anywhere from 1 to 2 hours, depending. Sometimes it could actually take longer. But in total, the results are available that next morning, and the total time, both hands-on and hands-off, is somewhere in the neighborhood of about 10 to 12 hours. And that's to do the sequencing test. So that's after Joe's piece, when he hands us off a specimen that has a low activity on his assay. Thinking about this in timeliness, sometimes we have to figure out how that's all going to work in the grand scheme of reporting our turnaround time. Now, something else that's come down the pike lately, we had a meeting in early October with our colleagues from North Carolina, from Duke, and Dr. Bali was there from the laboratory at Duke, and she had mentioned to us that there's this common exon 18 deletion. So Erin has just set this up. We haven't yet started reporting it because we have to get it approved, but it's actually a deletion of exon number 18 in the gene, and she set up what's called a gap PCR for this. So you can set primers up so that if you get this piece here under number 1, you have 902 base pair products, and that tells us there's no deletion. If we have a 902, which is kind of faint on here, and the 536, that's telling us this child is heterozygous for this deletion. And then if we get a sample where only that bottom piece is present, that's telling us the child is homozygous for that deletion. Now, according to Dr. Bali, it's quite common. From what I could find in the literature, it's in about 5 to 7% of alleles from people from the Netherlands who have Pompe disease. And so that's an appreciable frequency. And Erin has run through all of our carrier referrals to see if anyone had this mutation. And so far, we haven't detected it. But it's going to be part of our analysis that'll go on around at the same time that the sequence is being done, once we get it all underway. And Albany has a big Dutch heritage, so maybe we'll find an Albany child with this variant. So that's how we set up Pompe, and that's sort of how it runs today. The interesting thing at the end is that we have found several of mutations that have pathogenic, known pathogenic mutations. And this data has been sliced and diced different ways by Joe, myself, and Aaron in various talks. But we use two databases. One is actually in the Netherlands at Erasmus University, and the other one is at Emory in Georgia. So there's two databases that sort of catalog variants that help us and determine whether or not they are disease-causing. And you can see some are listed as very severe, some are less severe, some are potentially mild. There's all different classifications. So these are the easy ones because they've already been reported in people who have Pompe disease. And you can see the common mutations that people have talked about up here. These are the tough ones because these are the ones where we find them and we don't know what to really say about them. So we can run through a couple different software programs on the computer. One's called SIFT, one's called Polyfin, and what it does is map the variant to the protein to try to determine whether or not it is pathogenic or not. So we do a literature search, we use these software programs, we look at these databases to try and determine whether or not we can make a call whether this is a benign mutation or if it's a something that's very could be severe. So it gets a little bit tricky, and there's more. We find some novels. We've found several novel mutations as well. So hopefully as the knowledge base increases and more people do this, and we all work together on this, we'll be able to make better calls on some of these variants that we're finding. So I wanted to switch gears and just show you kind of where we are with mucopolysaccharidosis. You've heard it's an enzyme deficiency of alpha-1 iduronidase, the lysosomal storage disorder. It's called mucopolysaccharidosis type 1 because there's several of them. And the enzyme is responsible for breaking down these glucose aminoglycans, a compound in the body that you've heard about that normally get degraded in the lysosome. And two of them that are important for MPS I are listed here, dermatan sulfate and heparin sulfate. So that's Dr. Hurler up there. She doesn't look real happy. She was a German pediatrician. Interestingly, her husband was a vet surgeon, an animal surgeon, and her daughter also became a physician, and she was from Germany. Her son was actually killed in World War II. So in 1919, she described a syndrome of the corneal clouding and dwarfing and skeletal dysplasia, and it took her name, Hurler syndrome. And the frequency is listed at about 1 in 100,000. In 1962, below there, that dashing man is Harold Shea, and he was an ophthalmologist. And he had patients who had some symptoms that resembled Hurler syndrome, but it was milder. And he found them mostly through the corneal clouding, through the eyes. So he declared this an alternate form of MPS I, and it became known as Shea syndrome. In the old days, people named everything after themselves. We don't do that as much anymore. And then the intermediate one you've heard about is called Hurler-Shea. And Shea used to be its own MPS 5, and that's why if you look at the MPS literature now, you don't see a 5 because we call it kind of 1A or 1B. Dr. Shea was kind of interesting. He grew up in South Dakota. He was born in 1910, and he was schooled in a one-room schoolhouse in North Dakota where his parents moved to acquire some land. And he actually was a World War II veteran, and he retired as a brigadier general in the Army. And he was credited with saving the eyesight of Earl Mountbatten. So he's quite a famous guy outside of being an ophthalmologist. And he actually started the Shea Eye Institute, which is right there in Philadelphia where you all are. So it's still in existence, and he died in 1990. So we have Dr. Hurler, who doesn't look real happy, and Dr. Shea, who looks like quite a handsome man. The gene here is on chromosome 4. And for those who like the detail about genes, it's about 19,000 base pairs. There's 14 exons and 653 amino acids. And many mutations have been reported in this gene. The majority are private, but a few, as you heard earlier, have frequency. These are the two that were discussed earlier today. And so you can appreciate that the MPS gene is right here at the tip of chromosome number 4. And when you look on the computer, you can see also all the other genes that are around it. So for those who like to memorize these. But the interesting thing is it's right near the Huntington's disease locus as well. So, do you like that? Lisa D'Antonio in our lab has been working on this in her spare time. So she has set up, and this is about as far as we've gotten with this, but she's actually developed the primers. She's setting up these 8 PCR reactions to encompass the entire gene. And again, you can see we do everything the same way. We try and make as few fragments as possible to encompass all of the genes, various sizes of the amplification products, and she's actually done a real good job with trying to get this cleaned up. The only one that's tricky is this one here where you see these little bands. We don't like that. We like to see them a little bit cleaner because there's a lot of G's and C's in this gene, guanine and cytosines. They tend to be very sticky and hard to amplify. So we're to the point now where she's got the PCR product, and she has, we're kind of in our infancy of doing this right now, but that's about where we are. And you've heard, this is data from the Missouri program, I believe it was Missouri, yeah, that does, the Greenwood Center does their sequencing, and these are the same variants you've heard about earlier with the possible pseudodeficiency alleles, quite common in African Americans, interestingly. And we think that we're going to, because we're going to sequence the gene, we'll get a good sense of how many of these deficiency alleles are out there, and if we have the same proportion as has been discovered in some of the other lab right now that's doing it, 2 other labs that are doing MPS1. So with the sequencing test, we'll be able to hopefully send those kids home with a negative newborn screen after we get the DNA analysis completed. So I think that's all I have. I'm happy to answer questions, and I just wanted to thank Erin and Lisa for helping me put together a lot of the information on those slides and help me take pictures of the robots and that sort of thing. So thanks very much.
Just a quick, if anyone has questions for Michele, you have to get up from your seat and go to the mic, or else she won't be able to hear you. So are there any questions?
Okay, thank you for a very nice talk. Just a couple of questions. So do you have databases where you record the variants of unknown significance that if one would be detected, you go back to it?
So tell her who you are so she knows.
Okay, so my name is Arig Elgarbari. I'm one of the physicians at Children's Hospital Pittsburgh, and I direct the lysosomal storage disease program there. Do you have variants of unknown significance databases that you go back to, or do you just ask the different groups? I know that you've mentioned some, but the ones that have not been seen before, do they get saved somewhere where they're known, the enzyme correlation activity that is associated with it is stored there, where others can go back to it at some point in time? And then at the same point, do you have any data about the ethnic background? So not Caucasian versus like if they're from the Middle East or other country where the controls might be different.
Right. So we get, for the first question, we've been keeping track of the variants, and Erin's been very good about tabulating all of that information. We don't deposit it anywhere right yet because while there are variants of unknown significance today, we're not sure, and this is sort of Joe's topic, and the thing that we talk about a lot is these kids who have these variants, will they become symptomatic or not, and they're kind of out there. We've only been screening for Pompe a little over a year. So the goal of this is what we've done for Krabbe, and that is Denise Kay, who's right 2 doors down from me right now. She's put together a lot of our information on Krabbe disease and put together a database that we are hoping to be able to push out to the newborn screening community with the help of APHL in the next little while, we just were talking about the other day, and it would serve the exact purpose you're talking about. I know that there's other, there's ClinVar and there's some of these other databases out there that we sort of have to make a call as to whether it's, what the mutation actually does. And so we're coming at it from the opposite direction of the clinical world. And so at some point we're hoping the two merge. But we're happy to share any information we have. We usually will tell providers, when we call our providers, at least in New York, we'll tell them we've seen this before. This baby had normal enzyme activity on diagnostic tests and that sort of thing. So we're in the process of being able to do that, but we're not there yet with Pompe. We used to get information on race, ethnicity with our newborn screening collection forms, but we realized, well, I guess we kind of knew it, but we got to the point where we were basically relying on someone at the nursery to look at mom and look at the baby and make a call. I don't think they asked, the mother what her background was. So what we've turned around to try to do is on the referrals get the information when the provider actually sees the positive cases. We use the same cutoff right now for all of the newborn screens. But you do definitely see, I know in CF we see a higher proportion of babies with elevated IRT that are negative for CF. They tend to have higher IRT values, and I think there's been a couple reports about that. And now we have these pseudodeficiencies that we've seen, and there's common mutations in African Americans. So there's a lot out there, but we typically get that info back from the provider when they see the family.
So another question is, so were you considering the possibility of deletions and duplications if you didn't find anything on sequencing? I mean, you did mention the common deletion that was in the Dutch population.
Yeah, we are. So for the Dutch deletion, we're doing that one. We're not doing a global deletion and duplication, looking at potential others out there. We look at, for Krabbe, there's a common deletion that's highly pathogenic and is for infantile Krabbe disease. We look at that one. So we look at sort of the common or known ones, but we don't do an MLPA for every baby. We're looking at doing cystic fibrosis, looking at the full gene, and for that we're going to do the common deletions. But we just don't have right now those, sort of an array type of an assay set up to look globally at each gene. We always raise that as a possibility with the provider, and then they can send that out for that second test if they suspect that the baby is not truly a carrier. But right now we don't do that here.
Anybody else? Thank you, Michelle. We're all set.
All right.